For those of you who see me talk before, I'm afraid some of this might be a bit of a repeat of what you've actually heard already, but hopefully there's a bit of new information in there as well that you might not have come across. And for those of you who haven't heard me talk before, hopefully you will enjoy this. So I'm going to be talking today about my PhD research, particularly looking at the elements of citizen science that I use within it to find out more about slugs in British gardens. I've actually just completely finished my PhD and got one publication coming out this month. So there should be a bit more information coming on the horizon as well. But a lot of what I found is actually included in this presentation. Okay, so I'm going to start by talking about what we actually already knew about slugs in Britain back when I started my PhD in 2018. And then I'm going to move on to talking about the Slugs Count project, which was a huge element of my PhD studies. And I'm going to finish up by talking about some things that we kind of didn't really get to explore during the time of my research. It's left a lot of unanswered questions. So I just want to highlight those as areas of potential future research. So for anyone else out there who maybe wants to work on slugs and hopefully feel a little bit inspired and start thinking about some of these questions. So why slugs? So my PhD was fully funded by the Royal Horticultural Society, which is the UK's largest gardening charity. So they're really into gardening. So why would they want to fund a PhD on slugs? Well, it's partly because they've always been a huge problem for gardeners. The RHS has a gardening advice service where members can write in and ask any gardening question. And slugs regularly appear in the top 10 most inquired about garden problems. So they create these lists every year, and slugs usually are at the top or sometimes move down to second or third, but usually one of the biggest problems they get asked about. However, as a society, they've started trying to move much more towards supporting biodiversity and gardening sustainably and recognizing that, not all slugs are the same. So they wanted to know more and more about what species are actually present in gardens and which ones are causing problems for gardeners, which is where my PhD came in. And what's kind of surprising is actually slugs aren't particularly well studied in the garden environment or garden habitat. Often there's lots and lots of published literature on agricultural studies. There's actually very little scientific study of gardens themselves, or very few that have actually made it to publication. On top of that, we've also got new species constantly arriving in Britain. So, for example, when the Amazing Guide to Slugs was created, that the research behind that increased the UK fauna by more than 20%. So suddenly we had a lot more species known to be established in Britain than before. So we knew that the fauna had already changed, but we just needed to evidence that in the garden environment. So we currently have around 44 species present in Britain, and some of these can be quite challenging for beginners to identify. And generally, when you talk to a gardener, they think of slugs as being a single organism. They don't necessarily think of the fact that there's actually different species with different ecologies and different diets. So there was kind of a huge gap in education there, which we kind of wanted to try and fill as well. And when you talk to the public, their opinion is generally this, they have a head end, they eat all my vegetables, and then they poop everywhere, and I really hate them. And there's actually scientific literature to support the fact that slugs are one of the most unloved animal groups. So there's these two studies from 1995 and 1994, where the slug was rated as less useful, less smart, and less lovable. But also they've reported that 10% of adults reported anxiety when they saw a slug, and 44% of adults reported disgust when they saw a slug in the UK. So we really know that people in the general public are not big fans of slugs. So we set out to kind of try and change this attitude, but also get a lot of scientific data along the way. So we went in with this attitude of anyone with a garden can help us learn a lot more about slugs. And it has resulted in two citizen science projects that I ran. The RHS Cellar Slug Survey, which was open to absolutely anyone to submit records via iRecord, which looks for just a few target species. So I'm not actually going to talk more about this project today because I have talked about it in the past to the Society, and there's lots of recorded talks out there tackling this project. So if you want to know more, do look out for those videos. But I'm not going to go any more into that particular project today. Instead, I'm going to focus on slugs count. So this was a year-long study, which was limited to 60 participants. So we recruited 60 individuals and they took part in this project for a whole year, gathering data for us. And that's given us a year's worth of data on slugs in gardens. So this study was actually based, the methodology was based on that of Barnes and Weil from the 1940s. So Barnes actually studied 21 gardens in 1942 and 47 gardens in 1943, all of which were neighbouring in the town of Harpenden in Hertfordshire. So he came up with this methodology of searching at night with a torch, where he'd walk the same route each time and carry out nighttime searches, collecting any active slugs he saw. So any slugs that were crawling around outdoors. He did no searching or disturbing of foliage. So that meant he didn't turn over any logs. He didn't look under any flower pots. He didn't enter any sheds or outbuildings. It's just purely looking at what was active on the surface. So no digging or anything like that. He also experimented with surveying at different times and successive nights in the garden. So he's created this huge amount of data on what slugs were like in the gardens during the 1940s. And this was kind of the last published study that I could really find from that time period, though there was a smaller follow-up done by someone else in the 1960s, which also gave us a little more data in comparison to Barnes and Weil, but kind of focused on a slightly different area. So Barnes and Weil was kind of the main one where they actually looked at the full suite of species and identified everything that they found and figured out what was actually present in the garden. So we decided to take this methodology and then scale it up to make it a citizen science project. So all of the work that Barnes did, he did himself with the assistance of Weil as well as a statistician. So it was all just one man kind of doing this big project across multiple gardens, whereas we took the opposite approach of having one person in their own garden carrying out a search for us. So we set up this methodology and we put a call out in the press to ask people to apply. And we only had 60 spaces to fill because of capacity. So we knew that 60 was the maximum people we could handle. And we thought we'd really struggle to get that because, like I mentioned already, people hate slugs generally. They don't like them very much. So we thought, well, who's going to want to take part in this kind of project? And we're completely overwhelmed by the interest levels. So we had just under 3,000 applications, which was incredible because it then allowed us to narrow down to our 60 using geographic information. So we particularly wanted to get a really good geographic spread that was representative of the UK as a whole, excluding any islands. So unfortunately, anyone who lived on an island was excluded because we wanted to stick to the mainland to avoid any kind of geographic differences between island effects. And we managed to get this really, really good spread. There are a few little places in Scotland where we didn't quite manage to get someone involved, but generally it's been a really kind of nice grid pattern. And we're really excited because, this was going to be the first real in-depth study of slug species in gardens since the 1940s. And we were asking people to survey for an entire year, so from October to October, and asking them to go out once every 4 weeks using a standardized method. So we worked with them to part design this. So we set the search time of 30 minutes. We gave them the torch to use, so they were using the same light source. We provided various materials, so a lot of things were standardized. But we also had to work with the individuals themselves to understand their gardens and help them choose an appropriate route. So recommending that they try to choose a route that covered a full range of different kind of features or microhabitats within the garden. And I think that's one of the key things about this type of study is that gardens are not a standardized habitat. They are very much influenced and managed in different ways, but I'll talk a bit more about that later. So once those people had gone out to collect their slugs, they then brought them inside and spent some time using the Slug ID guide to actually identify them to species. And then the exciting part for me was they then put them in the post and sent them to the lab for me to verify. So they were sending live slugs so that I could then identify using the characters in the key, but also Royal Mail, the post carrier in the UK, doesn't allow ethanol to be posted. So it was kind of a win-win for me, really, in that I was receiving live slugs, which made them easy to ID. But also it was the easiest way to get them through the post system because they do allow the postage of live invertebrates. So from this year of study, we managed to find 32 different species of slug and collect just over 20,000 species of, 20,000 individual slugs. And we managed to actually have 59 people take part because one person dropped out before the survey started, unfortunately. But we're really pleased with how much material we actually managed to gather, which gave us a huge amount of information about slugs. So one of the first things we kind of looked at was at a geographic scale, looking at ecology and diversity. So we particularly wanted to look at the effects of weather. This is where our study diversified slightly from that of Barnes and Weil. So with Barnes, he actually selected nights that he thought would be good for slugs. So he kind of chose warmer, damper nights when he thought slugs would be more active, which I felt kind of biased the data a little bit. Whereas we decided to just also ask people to go out at in any weather condition, so that we got a full level of information about slugs activity across all weathers. But also I felt there was a risk that if we asked people to target a time they thought, like a weather condition they thought would be good for slugs, that's quite subjective and it might be difficult for them to judge, particularly as the people we involved had no background in marine ecology. They hadn't looked at slugs before. So to assume that they would know what would be good weather for slugs or to advise them of that felt a bit dangerous. So we just asked them to go out every 4 weeks, no matter the weather. So this gave us some really useful data on weather conditions, and unsurprisingly, it showed that temperature is quite a good predictor of slug abundance, with the higher temperature meaning that there's more slugs active. However, what was really surprising is that we had some incidents where we had slugs active in below freezing air temperatures, which is highly unexpected because generally the advice given is that if it's below 5 degrees Celsius, you will have much reduced slug activity or no slug activity at all. Whereas we had conditions where it was -5 and we had over 20 slugs found. What was interesting, though, is when you delve into those observations, so we talked to the citizen scientists themselves, we asked them about these findings, and it turns out in this case, when it was -5, they were found on a south-facing wall and it had been sunny during the day. So actually they had been able to use the thermal gain of the wall to actually be active on that surface. And I think that's just a lovely example of, how gardens contain a lot of microclimates and a lot of microhabitats, which means that there isn't a one-size-fits-all kind of rule in terms of when slugs can and cannot be active. What was also quite surprising for us to find is that there were no strong geographic trends in species diversity. What we were kind of expecting to see is that slug diversity would be highest in the south of Britain, partly because, it's warmer temperatures and that's where a lot of invertebrate invasions start from. But actually, when you look at this map here, the darker dots represent the highest diversity and the white circles the lowest. And you can see that these are completely randomly distributed. There's no real pattern to them at all. And again, this probably comes down to the fact that every garden is slightly unique and might contain different soil types, different moisture levels. They even have different rainfall patterns in those areas. So I think, again, you kind of have to look much more at bigger data sets to actually try and unpick this in future. So that was kind of at the larger scale, but when we looked at the smaller scale, we also asked our participants to collect a lot of data on the size of their garden, the age of their garden, areas of different habitat types or different what we refer to as garden features. So that could be things like mowed lawn, gravel paths, flower beds, vegetable patches, all kinds of things. We tried to cover as big a range of garden features as possible. We also asked them about management style, so whether they intensively managed their garden or kind of had more of a wild approach to that. As we've heard already, some people do like to embrace more of a wildlife friendly kind of approach to gardens, allowing more vegetation to grow up. So we then looked at the area of these different habitat features against slug diversity to see if there's any correlations or trends, and we found absolutely none that was significant. It seemed that there was no effect, though this may also be due to the sample size because it's a relatively small number of gardeners that we had in our study. And I think the level of variation between them is just probably bigger than we could possibly measure. And it is quite difficult to categorize habitats within a garden. So once we moved away from the approach of looking at the size of these features and then reduced it to a single count, so counting each feature as had a value of 1, we suddenly found that actually this had quite a significant effect on species diversity. So the gardens with the highest number of features within them actually had also the highest number of slug diversity. We also found that older gardens had larger numbers of native slugs in them as well, which is kind of interesting. So it may be that older gardens are less likely to be invaded by non-native species. But the biggest finding really was that the slug fauna in gardens in the UK is strongly dominated by non-native slug species. They are very, very dominant. So these are the 10 most abundant slugs found in our garden. Interestingly, number 1 is a non-native species that established in Britain after the 1940s. And in fact, as I've already mentioned, the most dominant species tend to be non-native. So 5 out of the 10 are non-native species. And what's quite interesting is Drossus reticulatum, one of our native species that's considered the most widespread and one of the most abundant slugs, actually was only 5th in this table. And what I don't have up here as well is the number of sites that these species also occupied. So the tramp slug, the Drossus maiden, is at the top there. Not only was it the most abundant slug, but it also occupied the most number of gardens as well. So most sites recorded this species. So the symbols, the little leaf symbols here also reflect the pest status of these species. So orange means that they are considered minor pest, red that they are considered a major pest, green that they're not considered a pest at all because they don't feed on live plant material, and the blue means that there's not enough information to actually make an assessment of their pest status. So in the case of arent flagellus and arent owenii here, this is because they are relatively recent introductions, and we actually don't know enough about their ecology and feeding behavior to actually know how much damage they're doing in gardens, which again is a really interesting point because they've made it into the top 10 slugs found in our garden. So there's obviously a bit of a knowledge gap there. So when we compare these findings to that of Barnes and Weil, what's really interesting is that 6, I think it is, 6 of the 10 species here with the red crosses over were not actually recorded in their study at all. And this is partly because they were either introduced later than 1940 or in the case of arent distinctus down there at the bottom in number 10, which is actually a native species, it may be that at the time it was aggregated with arent hortensis. But again, it just shows that there has been quite a large-scale potential change since the 1940s in the slugs found in our gardens. So what we can say from this data is that species found in British gardens has definitely changed since the 1940s, since Barnes and Weil did their study, with non-native species becoming increasingly more dominant. But because of the introduction of so many non-native species, actually species diversity in gardens has increased, which is kind of interesting because it's almost a reverse of what concerns other species groups have about declines in biodiversity. So we're actually seeing increased biodiversity in slugs, but that is driven by the introduction of non-natives. What was also kind of fascinating, though, is that we observed that the numbers of slugs in our garden has actually decreased. So when we looked at abundance versus how many Barnes and Weil were finding, our abundance was actually much, much lower, which is quite interesting. Again, there's some caveats to that because obviously Barnes and Weil were being very selective about when they actually went out and surveyed. So they were choosing what they thought was good weather for slugs. Therefore, it's possible they were finding higher abundance because of that. But it's also possible that the way people have gardened has changed dramatically. So it's worth remembering that in the 1940s, we've just come out of the war. People had been digging for victory, growing lots of vegetables. So there might have been a lot of differences in the way that gardens were managed at that point. Whereas now we're seeing a lot more people paving over gardens, tarmacking over gardens, putting gravel down. So maybe that's not as slug friendly as gardens used to be. But there could also be the fact that this was before molluscicides were widely used or introduced. So again, that could also be one explanatory factor for why abundance of slugs is potentially lower now. And what's interesting is that this is both the case at local and national level. So it's worth remembering also that Barnes of Wild study took place in a single location in Britain. So they looked at just one town, whereas we were looking at a really large geographic coverage. So again, that could explain some of the differences. So what we actually did was we subset the data from Barnes and Weil to their most studied gardens, because they weren't always consistent as to which gardens they actually studied, and also subset our data for the nearest garden to that locality, the nearest 3 gardens as well, actually, at one point. And we compared the data, and they showed very, very similar results. So they showed also that abundance was lower, but also their diversity was much higher, and they contained lots more non-native species. So interestingly, there are a few species that maybe aren't doing so well. So this is particularly one of them, Tandonia budapestensis, which is also a non-native species and considered a major pest of root vegetables. So in the 1940s, it's actually represented 21% of all the slugs that they found. So it was really, really abundant. Whereas in our study, it represented just 1% of the slugs that we were actually finding. So much, much lower abundance. And that's really interesting potentially, but it could also again be explained by changes in the gardening style since the 40s, particularly if in that area they were growing a lot more vegetables. Vegetable patches were actually mentioned quite a lot in the paper that Barnes and Weil published. So that maybe could explain why we're detecting less of them, because maybe we didn't have as many gardens in our study that were actually growing vegetables, which this is a major pest of root vegetables. Therefore, you'd assume that it would do better in gardens with root vegetables growing. However, it's one of those species that maybe it could be worth looking more at the historic data of, so the records that the Society holds, and the ones on the MBN Atlas, to see whether this is actually a continuous trend or whether it's just an unusual finding in our study. There's also some species that haven't really changed much at all. So arent circumscriptus, in our study, it was represented a very small proportion of species, and it was the same in the 1940s. The leopard slug, again, relatively similar between the 1940s and the 2020s. And the yellow cellar slug, also relatively similar in terms of abundance in both studies. Though this study also did generate some new information on distributions, particularly some information on less recorded species. So in this example, Tandonia cristata, we found this actually in 21 surveys across 6 gardens, and 5 of these were new sites for the UK. This is very much a non-native species that's a fairly recent introduction. What's interesting was this showed that it was possibly even more widespread and common than currently is known. It's also interesting that we didn't find it outside of those 6 gardens, so they are all kind of limited geographically to within relatively close to the area where it's previously been recorded. So this could suggest that, you know, it's not spreading further north easily. So maybe it's restricted by environmental factors, possibly. But yeah, it's just really useful to have a bit more information about the spread of this species. And it was kind of exciting and interesting for our participants to find this out as well. So I've talked about the slug content of our research, but I just want to also talk about the human element, because that was quite a large part of the project, was actually understanding the people involved and what impact the project had on them in terms of identifying and recording slugs. So when we selected people for this project, we intentionally tried to select some people who had a little bit of experience of previously identifying slugs, but also including a lot of people who had 0 experience before. They hadn't tried, and they hadn't really had the opportunity to look at slugs much before. And this was partly because we wanted to measure how they did in terms of slug identification. So because we had a broad range of different people coming into the project, we provided a lot of training at the beginning, particularly on using the key and identifying slugs to species. And we did this via online webinars because unfortunately this was during the COVID restrictions. So we weren't able to meet up in person until way after the end of the project. We also did provide some update webinars as well online. And we made sure that all those recordings were accessible to participants. So those who couldn't attend the live webinar due to various activities were able to actually go and watch those recordings back. We also provided individual emails to every participant on their slug identifications every month. So we made sure to give them a list of what they said they recorded in the previous survey versus what we actually received in the lab. And this gave them a chance to kind of look at where maybe they've gone wrong and try and improve. And often we'd try and leave a little comment as well about some of the species they might have confused to help guide them a little bit. I think this really engaged them and kept them motivated because we had quite a few participants during the project say how difficult they were finding it and how, they just wanted to give up because slugs are really hard. But actually, we were really, really pleased that we only had 2 people out of 59 leave before the year of surveys ended. So we managed to retain them quite well. And despite, everyone having busy lives, we managed to actually complete 93% of all the scheduled surveys as well. So we had a really good high level of engagement in generating lots of data. So one of the key things I wanted to look at was how accurately people can actually identify slugs as citizen scientists with relatively minimal training. Because this was always been highlighted as potential challenge for this project. And because of the way we'd collected the data, we were in a unique position to be able to do that because we'd asked people to identify them at home, send us a list, but also they'd then posted the material in, we verified that, and then we had basically 2 data sets that we could compare. What was interesting was that participants generally overestimated slug abundance and species richness. Now, the overestimates of abundance, I think, could be explained by the methodology, and because things were being posted, we did suffer some losses in the post. It was not unusual for a package to arrive with fewer slugs than it had when it set out. So we were getting people to write the numbers of slugs in each box as they sent them. So we were able to kind of observe that. However, we did also notice there was a few times where participants undercounted. So they would say that there were so many slugs in a box, and when we count them, we'd find there were actually more. So which is just human error. So that could also maybe slightly explain that. In terms of the estimates of species richness, that was quite an interesting one. What I did observe was that often people would report finding quite a rare species. So, for example, we had a few reports of Limex cinnamoniger, which is an ancient or old woodland specialist. So you wouldn't necessarily expect to find it in the garden. When we received them, most of the time they were leopard slugs. In fact, we had none that were positive, but sometimes there were maybe other species. And I think that maybe there was a keenness to find something rare or unusual, that maybe citizen scientists are often quite keen to find something a little bit unusual. So maybe that's partly what drove that. Maybe it's human behavior, but it might also just be them getting confused in the key, or some of those species do have slightly similar characters as well. So it's quite a good learning opportunity for understanding perhaps where people were struggling. So I wanted to know how people felt they did with Slug ID, because we knew that Slug ID was hard, but no one's really quantified that before. So we're trying to do that. So we asked participants to tell us before participation what they thought their level was, and after participation what they thought it was. And the majority of participants recorded significant improvement in Slug ID experience. So you can see from the graph here on the left, before participation, there's a really huge purple bar on none, so they had no experience at all. And after participation, that's all shifted. So everything's shifted to the right quite a lot. The one exception to that is the expert category there. So at the beginning of the project, we had 2 people who considered themselves expert, and by the end of the project, we had 1 person who thought they were an expert, or is an expert, I should say. And that was a reflection of maybe people overestimating their own ability. And I think that's always quite a hard judgment call to make, is when someone asks you to define your experience level. It probably depends on your personality or various factors, how you classify yourself. So there are certainly some people, I think, who thought they were beginner level, who maybe had experience in other groups, so actually were more experienced than they thought. Anyway, so generally people felt that slug identification was challenging. So we asked them to compare it to their understanding of other scientific topics to see how it compared. And generally they found it more challenging than understanding the science. But thankfully, 95% of participants enjoyed taking part in identifying slugs, despite the challenges. However, we did find that there are lower levels of interest in continuing to identify and record slugs outside of the project, with only 66% of people responding positively to that, which I still think is a good result, because it shows that we've still got a huge amount of potential recorders out there who are willing to go and record slugs. But it does, I think, reflect how difficult slugs were, and also people's motivations for taking part. So a lot of the motivations we talked to people about, or people reported, was that they felt they were contributing to a scientific project, whereas I guess they didn't necessarily see recording as doing that, even though I would argue that it very much is still contributing to our scientific knowledge of the slug fauna. So that was based on people's opinions, but what we also wanted to look at was, can we actually prove that using quantitative data as well? And one of the things we did was, at the beginning, we'd asked them to classify their category level of experience, and we actually grouped people by that to then look at the proportion of misidentified material. So what this graph shows is, at the bottom, you've got the survey number. So this starts from survey 2, because we decided to discard survey 1 data due to some changes in the methodology. So that kind of represents time, I suppose, because each survey you're adding a bit more experience. And then on the left axis there, you've got the mean misidentification score. So that's what proportion of misidentifications occurred in that survey for that group. So what ideally we'd want to see is everyone on 0, but we knew that's not realistic. Not everyone's going to get everything right all the time. But what you actually see is that the mean misidentification is around 50% of most of the time. So that meant people were misidentifying things about half of the time. And what we fully expected, actually, was to see that the more experienced group here in orange would start out really accurate and stay that way throughout the project. And the groups of beginners, or those with no experience, which are the the pink and the green on this chart, would start quite low. So start really high with high levels of misidentification, and then over time reduce down to being much more accurate and get more on a level with the more experienced group. But what you'll notice here is actually it's a complete mess. All the groups constantly overlap. But overall, there is actually a bit of a significant downward trend. So over time, everyone was getting more accurate. But what was really interesting is there was no significant difference between prior experience groups. So that meant that people who'd had experience identifying slugs were no more accurate than those who had no experience. And actually the improvement in accuracy was very much driven by the number of surveys done rather than the prior experience they had. So as I've talked about before, there's probably some explanations for this to do with the way that people were self-defining their own ability. You might have some people who'd studied different groups before and picked up slug identification really quickly, but therefore put that they had no experience of slug identification, though, because they'd not tried before. And you might have people overestimating their ability as well. So those are the findings of our project. I'm now going to talk a bit more about things that we didn't really get answers to, but have come out as things that maybe need a bit more investigation in the future. So one of the key things, of course, is the fact that non-native species are now incredibly dominant in our gardens. And this got us thinking about, well, where are these actually coming from? So I think we need to do a lot more to understand more about the vectors in the introduction and spread of non-native species, whether that's things like the horticultural trade, plant nurseries, or even just hitchhiking on traded commodities and shipping containers. We really have very little information in the UK of how new species are actually arriving and how they're spreading within the country and what measures we can take to prevent this. So I was lucky enough to actually get a bit of funding from the Department for Environment, Food and Rural Affairs, so DEFRA for short. And they gave me a future-proofing plant health grant to actually carry out a review to try and identify species with potential to arrive in Britain and be a threat to plant health. Also looking at the different corridors that they maybe are using to travel. This is now publicly available online, so do feel free to have a read. We've suggested some species that we think could arrive here next, so they might be worth looking out for. And that does include a few slugs. But what I think is also quite interesting is this is not a unique problem to the UK. I think further afield, other countries as well, they're not doing much monitoring of what slugs are arriving and how they're getting here either. So that might mean that this review doesn't include all those species that could arrive within the next 10 years or so, because they might not be on our radar yet. One of the other challenges we had was identifying cryptic non-native species. So for this study, we didn't actually dissect any material. We relied solely on external characters for identification. So because this was realistically what citizen scientists could do. So we didn't ask them to then start dissecting slugs for us. We did actually collaborate with FSC Biolinks to run a few workshops where we got other people who hadn't been involved in slugs count to come in and start dissecting material for us to see if it was feasible to get them to do that, because we had 22,000 slugs potentially to go through. So there's no way the one person could do that. But you can see from this photo at the bottom here, the turnout for that was quite low. So I think there's a long way to go to get the public engaged in that kind of discovery. But there's really key important questions here. You know, can external features be reliable for separating these cryptic non-native species? And I know that we've got a great talk coming up with ConchSoc on this Ambegalimax group as well. And I think for that group, the answer is probably no. But then how can we ask members of the public to be able to record them if they're not willing to dissect, if external features are not reliable? So there's quite a big thorny issue there to talk about. We also know that there's a lot of hybridization happening between slug species in Britain and across Europe as well. It's quite important for the public then to understand which ID characters are actually reliable, particularly as it's that large arenator aggregate group that often hybridizes, and it's resulting in these kind of very overlapping colour forms. And those are the groups that are very widely encountered by the public. So when you get casual recorders on platforms like iNaturalist and iRecord submitting photos, it's often these species, and we can't really take them beyond aggregate, which makes it quite, I guess, discouraging for new recorders, because, you know, the really common species they're seeing can't be reliably ID'd. So it'd be really useful if we could actually find ways of distinguishing between hybrids and natives if possible. It also seems like hybrid species are potentially becoming more common in the UK. And what I think gardeners would really like to know is this a problem? Is this going to change the damage they do in gardens, particularly when you talk about native species Arent rufus hybridizing with non-native species Arent vulgaris, which is considered much more damaging to plants. There's also not great understanding about the ecology of some native slug species. There's some species where we just don't know enough about their life history traits. We don't understand what they eat. And we have very little understanding of the impacts they potentially do on their ecosystems. So, for example, the green cellar slug is really widespread and very abundant in our gardens. It was very high in those top 10 species that we found. It's a detritivore, but is more detritivores actually a good thing for our gardens and wider habitats? That's something that's not really been well studied, and something that potentially we need to know more about. So thank you very much for listening. I hope that was an interesting talk, and hopefully you've got some really interesting questions.